Welcome back everyone, I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about the G6PD deficiency and how does it protect us against malaria. Okay. So as you all know, the G6PD enzyme or glucose 6-phosphate dehydrogenase enzyme is the rate limiting enzyme of HMP shunt pathway or pentose phosphate pathway, where it converts glucose 6-phosphate into 6-phosphogluconolactone, and during this conversion NADP plus gets reduced into NADPH2. Okay. Then this 6-phosphogluconolactone gets converted into 6-phosphogluconic acid with the help of hydrolase enzyme, and then this 6-phosphogluconic acid with the help of 6-phosphogluconic dehydrogenase gets converted into ribulose 5-phosphate, and during this reaction also NADPH2 is formed. So the main thing is that the G6PD helps in formation or generation of NADPH2 during HMP shunt pathway. And why this NADPH2 is very important for our cells? Because this NADPH2 helps us in free radical scavenging, mainly in our RBCs. Okay, now let us focus on how this NADPH2 helps in free radical scavenging. See, this NADPH2 generated during HMP shunt pathway actually acts as a coenzyme of glutathione reductase enzyme. Okay. Now, this glutathione reductase enzyme with the help of NADPH2 converts oxidized glutathione into reduced glutathione. Okay. And this reduced glutathione is very important for our antioxidative system. This reduced glutathione helps the glutathione peroxidase enzyme to convert toxic hydrogen peroxide into non-toxic water molecule. Okay. See, this hydrogen peroxide, which is a reactive oxygen species, is continuously formed within our cells due to continuous reaction of superoxide anions, which are free radicals. Okay. Now, if this hydrogen peroxide gets accumulated in our body, in our cells, it leads to the cell lysis. Okay. And in RBC, this cell lysis leads to hemolytic anemia. Okay. So we need to convert this H2O2 as soon as possible to H2O with the help of glutathione peroxidase. And the reduced glutathione helps the functioning of glutathione peroxidase, whereas the reduced glutathione is formed by the reaction of glutathione reductase by the enzyme glutathione reductase with the help of coenzyme NADPH2, and this coenzyme NADPH2 is formed due to the activity of G6PD in our HMP shunt pathway. So that is how G6PD helps in free radical scavenging of our cells and saves our cell from the toxic effect of hydrogen peroxide or reactive oxygen species. Okay, now I think up to this portion it is clear in your mind. Now let's take a look how this G6PD deficiency occurs and what happens in the malaria prone area due to this G6PD deficiency. See, G6PD deficiency is a X-linked recessive trait. Okay. So, in those people who have deficiency in G6PD enzyme, okay, they have obviously the reduced activity of HMP shunt pathway. As a result, the level of NADPH2 in their body is also reduced. Okay, and if the NADPH2 level is decreased, as a result, what will happen? The activity of the glutathione reductase will also get decreased. Okay. When the action of glutathione reductase is decreased, obviously the amount of reduced glutathione formation is also decreased. Okay, and as we know, the reduced glutathione is very important to convert the H2O2 into H2O and to save the RBC from the fragility. So during the decrement of reduced glutathione, obviously the RBC fragility will increase, okay, and which is not so good for our cells. But nature has played a very intricate role with those people who have G6PD deficiency and who lives in the malaria prone area of Africa, South Asian countries and Middle Eastern countries. Okay, we can see that the G6PD deficiency has a very high frequency in these countries. Okay, and this G6PD deficiency offers resistance to the plasmodium infection, which is the primary cause for malaria in these countries. Okay, now you may ask me how G6PD is offering the resistance against plasmodium. See, like many other parasites, plasmodium also do not have any antioxidant system. Okay, so when they infect the RBC of our body, our RBC is already G6PD deficient. As a result, the amount of H2O2 or the reactive oxygen species is very high already in our RBC. And now the plasmodium is affecting that RBC. Okay, now what will happen? Plasmodium itself has no antioxidant system, and G6PD person has also have the reduced antioxidant system in its body. So as a result, the plasmodium will be attacked by the reactive oxygen species and it will get dead. Okay, and it will be fatal for it. So it cannot sustain its life cycle in those G6PD deficient RBCs, okay, due to absence of reduced glutathione. Okay, and another thing is that also in the G6PD deficient individuals, premature hemolysis of RBC occurs, which decreases the number of available host cell for the plasmodium. Okay, so as a result, plasmodium has very less number of host cell to infect, and if it also infects, it cannot survive in those RBC due to the presence of high amount of H2O2 in absence of G6PD. Okay, so as a result, the life cycle of plasmodium gets disrupted and the people living in those malaria prone areas get saved from getting malaria. Okay, and that is how G6PD deficiency helps and protects those people living in the malaria prone area of Africa, South Asia and Middle East from getting malaria. Okay, now I hope this concept is crystal clear to you, and if you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe our channel for more such videos, and please comment down below if you like the video. Thanks for watching and happy learning.